Hi, welcome back to the Sound of Science. I'm Amy Myers, your host. Today we're going to be talking about the properties of biological molecules. One of the most important things that you need to take away from this video is that function and structure are tied together. So living systems are arranged into hierarchies, and you guys know this, right? We've talked about how cells make up tissues, tissues make up organs, organs make up organ systems make organisms, right? You know the hierarchies. Well, the structure at each of those levels can help determine the function either at the level or in the overall. And any change to the structure will change the function most of the time. Sometimes it doesn't, but most of the time it will. Biological molecules' function is absolutely determined by the structure. Now, we do have examples where we have molecules that have the same formula, but it's a different structure. So the example I have here is ethanol and dimethyl ether. And dimethyl ether is a gas at room temperature. Ethanol is a liquid, which is highly volatile at room temperature. And if you take a look at ethanol versus dimethyl ether, you can see that their formula is the same, C2H6O, but when you look at the structure of the molecules, it's vastly different. And so where the atoms are arranged is just as important as what atoms are present. Most macromolecules are going to be made up of monomers that get stuck together, bonded together to create a polymer. Mono meaning one, poly meaning many, right? Makes sense. So if we look at a nucleic acid, its structure is essentially a 5-carbon sugar, a phosphate group, and a nitrogenous base. That's what's going to make up its monomer. And then we call those nucleotides. The nucleotides, A, T, U, C, or G, are going to be stuck together into a linear strand, a sequence. This is how we're going to store our biological information. DNA and RNA are the two types of nucleic acids that we really talk about. And the difference between them is the sugars. There's a difference in the sugars, and that difference is literally one oxygen atom. So DNA does not have that extra oxygen atom, as you can see with the red circle in the diagram below. So it's deoxyribonucleic acid. It has a deoxyribose sugar, 5-carbon sugar that is missing an oxygen. RNA is a ribose sugar as the 5-carbon sugar, and it has that extra oxygen. Because of that, DNA is going to be double-stranded. It's going to remain in the nucleus. It is involved in replication and then is transcribed into mRNA. The RNA then can leave the nucleus because it is single-stranded. It's a smaller molecule, and it's used in protein synthesis. It's going to be translated into amino acids. All of this is stuff that you learned when you learned about central dogma and protein synthesis. But again, it's based on structure. So the lack of an oxygen changes the entirety of the molecule. Let's talk amino acids. Proteins are made up of the monomer amino acids. And we have about 20-ish types of amino acids, which you don't need to memorize. But just know that all of them kind of have the same basic structure. They have an amino terminus and a carboxyl terminus, and those are just the ends. And then all of them have an R side chain, which makes them different. So these amino acids have a specific directionality. They are put together in a specific way, and the order of the amino acids is going to determine the overall shape of the protein. Shape like structure. The structure will determine the function of the protein. So I've given you an example here of the protein structure. So take a peek, and you can see that the primary structure is just amino acids bonded together. The secondary structure is how those amino acids start to twist, and they actually form these things called beta sheets that sort of fold together. And those folded, twisted amino acid strings form the tertiary structure. Once you put that tertiary structure together with several other tertiary structures, you get what we call the quaternary structure, which is the overall globular protein. These are complex molecules, but it's all determined based on the amino acids that are present and the order in which those amino acids are placed. Those side chains that we talked about that make the amino acids what they are, those side chains may be hydrophobic, hydrophilic, or they can be ionic. And depending on their structure and what they are, determines how they're going to interact with the other amino acids and with the environment. And that is going to determine overall the shape of the protein. When the groups are hydrophobic, they can interact with other hydrophobic groups. When they're hydrophilic, they can interact with other hydrophilic groups. So now on to carbohydrates and lipids. So carbs and lipids both have the same kind of job, a little bit. They're both able to store energy. Carbs store short-term energy and lipids store it long-term. Lipids are, of course, also able to provide insulation. They're going to make up cell membranes. So it's very, very important. Carbs can also be used for structure. So they have multiple purposes, multiple functions. Complex carbs are going to be made up of monomers that have different structures, and therefore they can have different properties. This is going to be able to change the overall function of the carbohydrate. And so that's something to kind of be on the lookout for. Like, for instance, glucose and galactose. If you take a look, glucose and galactose have the same basic formula. The only difference is on the number 5 carbon there, because remember, we always start counting from the carbon, to the right of the oxygen. So it's that number 5 carbon, the one to the far left of both diagrams. If you'll see there, the hydrogen and hydroxide are reversed from each other, right? Their directionality has been changed. And as a result, we've gone from glucose to galactose. This is a pretty big difference. They actually behave differently. So that structural change, even though it's a small structural change, apparently makes a big difference. When we talk about lipids, lipids are nonpolar. So they're going to be made of subunits. They don't really have monomers. They are going to be made of things like fatty acids and glycerols. Fatty acids are made of parts that determine structure and, again, the function based on saturation. So I have an example there. You have saturated and unsaturated fatty acids. Saturated just means that every carbon is bonded to the maximum number of hydrogens it can have based on the number of carbon-to-carbon bonds. Unsaturated means there's a place where they don't have all the carbons, or all the hydrogens they can potentially have, and therefore the carbons have created a double bond. So anytime you see a double bond, you know that it's an unsaturated fat, okay? When we look at these fatty acids, their function is vastly different depending on whether they have any of these double bonds. Phospholipids, which make up membranes, have both hydrophilic and hydrophobic sections, and that controls their interactions. So it's a hydrophilic head, phosphate head, and it's a hydrophobic tail. So the fatty acids that are hydrophobic found on the bottom, and then the hydrophilic, the water-loving section, is found in the head of the lipid. So why am I making a big deal about the fact that we've got hydrophobic and hydrophilic sections, right? So the thing is that membranes not only have these lipids, but they also have proteins. And remember we talked about how the amino acids could be hydrophobic or hydrophilic? Well, when you fold the protein, it's going to tend to have the hydrophilic sections to the outside so that that can interact with that watery environment, because hydrophilic is water loving. Hydrophobic sections tend to stay together inside. And when you see the protein in the membrane, you're going to see that the hydrophilic sections are going to hang out with the hydrophilic portions of the lipid bilayer. And then the hydrophobic sections are going to hang out with the fatty acid portion of that lipid bilayer that's also hydrophobic. Hydrophilic can interact with each other and with the environment. Hydrophobic can interact with each other, but they can't interact with the environment, which allows the membranes to fit perfectly into allows the proteins to fit perfectly into the membranes. So here's a quick review. Structure determines function for macromolecules. The different ends of the amino acid, the amino and carboxyl terminus, will shape the protein itself. Monomers and complex carbs assemble based on the structure of those monomers, and the saturation in lipids will also help determine their function because it determines their structure. Polar regions interact with other polar regions in water. Nonpolar can only interact with other nonpolar molecules. Hopefully this has given you a good overview